So in the next few videos I will be doing very useful content for the MBBS students because I'm going to cover the bacteriology almost like lab diagnosis, because in every exam they'll ask in microbiology mainly lab diagnosis, and I'm going to cover the lab diagnosis of every bacteria one by one, like gram positive and gram negative bacteria. So I'll be doing a series of videos and cover almost everything about the lab diagnosis, and I have written in very simple language so that everyone can understand easily. So this will be mainly helpful for your second year exams for MBBS. So in this video I will tell only the basics of lab diagnosis. So what we'll generally see in lab diagnosis or what we do. So in the next video I'll start with the individual bacteria. So in coming to this video, this is about the lab diagnosis, generally what we'll do. So first we'll collect the sample. So how the sample is collected and where it is stored, or what are the techniques used for sample collection, we'll discuss here. And how it is transported. If it should be transported to a distance, then how it is transported, or which transport media we use, that is also we'll discuss. And in next, after the sample collection, what we do generally is we see under the microscope. So for seeing under the microscope, we use different stains like gram stain, AFB stain, and Albert staining, etc. So we'll discuss about these later because these are the basics, we'll know later. In next, after the microscopy, we do the culture, means will grow the bacteria. So we have different media, like it's better if you know this order, like if you can by heart it for 2 to 3 times, and you'll know this order, and it will be very easy once you know this order to write in the exam. So in the culture, generally we go in this order. First we try to grow in the nutrient agar, next nutrient agar slope. So next this is the blood agar. In the blood agar we have, it is an enriched media, and also we grow in Loeffler serum slope. See, I'm telling all the culture medias that are used in this bacteriology, like some of the bacteria may grow in blood agar or may produce beta hemolysis in the blood agar, and some may not produce the hemolysis. But I'm just telling what are the cultures used, like that is nutrient agar slope, blood agar, McConkey's agar, and liquid medias, that is peptone water, BHI, brain heart infusion agar, and glucose agar, and other selective media. Okay, and for some like Neisseria meningitidis, that is it causes the meningitis infection, so CSF examination is also done for that. And next, these are some of the test that is done. They are the biochemical test. If some of the bacteria produce toxins, we do the toxin demonstration. Next, we do the serology and molecular methods. In molecular methods, we know PCR is done mainly. So this is about the introduction for lab diagnosis. We'll go in detail about the lab diagnosis of individual bacteria in the next videos. And I promise you this video lecture series will be very helpful, and I'll make it short, okay, not so big videos, so just 5 minutes each or even less, okay.